and I'll speak.
Yes, sir, already sharing. The slide is visible. Yeah.
So much. Very nice presentation by Dr. Usha ma'am. Basically, most of the things dealing with rare diseases have been dealt with by ma'am. But I thought to speak a little bit about craniosynostosis because it is a very common presentation of many rare disorders. And in fact, craniosynostosis is itself a rare disorder. So little bit about me. As already told, I'm an assistant professor of medical genetics at the newly created Department of Genetics at Postgraduate Institute of Childhood, Noida. As you all know, as the burden, please wait till when I say next, then please change the slides. Oh, is that okay? Hello?
Yes, sir, okay.
Yeah. So moving on to the first slide, can you move next? Yeah. So basically, regarding the burden of genetic disorders, these are, though they are rare as compared to other common disorders, infection disorders and other things, but still around 3 to 5% of all live-born infants have a major congenital anomaly. And in different published studies, it was found that around 15% of all pediatric admissions have some underlying genetic defects. Around 20 to 30% of all infant deaths are due to genetic disorder. Interestingly, in mental retardation patients, around 50% individuals have some or other genetic etiology. So basically, rare diseases are very important and should be taken into account while dealing with the, especially the pediatric populations. Next slide. Yeah, now regarding craniosynostosis, as most of the clinicians who must have joined the session, they know it is the premature fusion of one or more cranial sutures, usually encountered early in life. The incidence ranges in different populations, and it has been found to be 1 in 2100 to 1 in 2500 children. Apart from that, a specific genetic etiology can be identified in around 20 to 30% of cases. Next. Yeah, so basically brain grows where it finds space. We all know there are sagittal suture in the midline and there are coronal sutures anteriorly and then the lambdoid suture posteriorly. Basically, any fusion of the skull in any one direction will lead to the growth of brain in the other direction, leading to the various deformational shapes. Next slide. Yeah, so basically the skull shapes depending on the suture involvement, like if sagittal suture is involved, then there will be growth of brain in the anterior-posterior direction, leading to a skull deformity called as dolichocephaly or scaphocephaly. It is the most common of the craniosynostosis, affecting a single suture, and generally 80% are male. Then if the coronal suture synostosis occurs, then bilateral if it occurs, it leads to a brachycephaly or the acrocephaly. Around 18% of craniosynostosis, there is fusion of the coronal suture. Then if there is fusion of the lambdoid suture, it leads to occipital plagiocephaly, which again encounters around 10 to 20% of the patients. Then again, metopic suture closure will lead to trigonocephaly, and if there are multiple sutures, craniosynostosis occurs, it leads to a pterycephaly or the oxycephaly, which appears like a tower skull. Next slide. Then basically there is a term called Klipps-Ladel or cloverleaf skull, which is a premature fusion of all the cranial sutures, and it leads to a life-threatening and requires urgent surgical management. Next slide. Yeah, so basically the classification of the craniosynostosis, it can be primary or secondary. Secondary we are not going to discuss today. Primary, basically it can be syndromic as a part of a syndrome with other manifestations, or it may be a non-syndromic craniosynostosis. Next slide. So basically here they have shown the approach to craniosynostosis. Normal brain growth, as you can see in the figure A, and when there is genetic abnormality of the suture maintenance, then it leads to a poor brain growth. And again, you can see sagittal synostosis is being shown. The skull shape is the typical dolichocephaly, and there in the other figure, the sutures are mentioned. And again, regarding the predisposition factors, environmental causes are an important entity. Valproate ingestion in the mother, twinning, all these things can lead to craniosynostosis. And then again, while seeing such children or patients, additional clinical features are important, which we'll discuss in further slides. Next slide. Yeah, now what is important when you are dealing with a child with craniosynostosis? History is very important, as in all other cases, a 3-generation family tree. And the history of in pregnancy in mother, the history of decreased liquor, abnormal fetal presentations, etc. And again, uterine abnormalities like bicornuate uterus, which may hamper the growth of the fetus. Then the precipitate delivery, skull shape at birth, or progression of deformity, all these things are important. And again, positional, it can be there because of this specific sleeping position of the baby. Again, most of these craniosynostosis syndromes, they present with developmental delay or intellectual disability. Hence, a developmental history is very important. And again, any seizure or signs of raised intracranial pressure due to the premature fusion of the sutures. While examination, which is very important part of any other genetic disorder, especially developmental disorders, the occipitofrontal circumference or the head circumference you should take, and other growth parameters. And again, the position of the fentanyl and size is important. We should palpate for the sutures to see their ridging, the skull shape, we should ascertain as seen in previous slides, and then ear position, shape of the face, supraorbital ridges, exorbitism, hands and feet, because they are important. We'll see in further slide, there are certain syndromes like Apert syndrome in which syndactyly can be there. Any cleft palate present or not, broad thumbs and hallucis, assessment of parental shape, and the fundus and hearing evaluation. Next slide. Now, what are fibroblast growth factors and receptors? These play an important role in human axial and craniofacial skeletal development. FGFRs are basically tyrosine kinase receptors that consist of an extracellular ligand binding domain, a transmembrane domain, and intracellular divided tyrosine kinase domain. Basically, FGFR 1, 2, and 3 are crucial for chondrogenesis and osteogenesis. However, FGFR3 has a more important role in chondrogenesis. And mutations in all these genes are responsible for various types of skeletal dysplasias as well as craniosynostosis. Next slide. Yeah, basically, we have shown in this slide, in this table, basically the various types of mutations and the disorder which are occurring. Like FGFR will lead to fever, osteoglyphonic dysplasia, FGFR2 will lead to other syndromes, similarly for FGFR3. Next slide. Yeah, in this we have shown the FGF signaling pathway, basically to show the role where these genes are involved. Next slide. Yeah, in this we have shown a prototype of the FGFR3 protein in which there are three important domains which are common to all FGFR proteins, the immunoglobulin 1, 2, and 3 domain. And then we have the tyrosine kinase part. The TKA and TKB are basically intracellular, while the IG1, 2, and 3 are the extracellular domains. And you can see these are quite a specific positions, and the mutations and the typical like arginine being replaced by cysteine S248 will lead to thanatophoric dysplasia type 1. So basically, next slide. Yeah, so basically I wanted to say that these FGFR genes show a highly conserved relationship between the mutations of a particular amino acid and resulting phenotype. Okay, next slide. Yeah, now FGFR related craniosynostosis, majority of mutations in FGFR2 localized to 2 exons encoding the extracellular immunoglobulin 3 domain. Similarly, both fever and Crouzon syndrome may be caused by replacement of cysteine at 278 position by phenylalanine or tyrosine. Apert syndrome is typically caused by a specific missense mutation in the highly conserved linker region between immunoglobulin 2 and 3 domains of FGFR2. Next slide. Now, basically about non-syndromic craniosynostosis. These account for around 85% of the cases, the genetic cases, and there is high genetic heterogeneity, and largely most of them are unexplored. Next slide. Yeah, in this pathway, we have shown the various genes which are implicated in the non-syndromic craniosynostosis. Around 20 set of genes are there. Some of them you can see highlighted in red. And specifically, I wanted you to see you for those having a star like ERF, TCF12, and TWIST1, because these are mostly involved in the familial non-syndromic cases. Next slide. Yeah, now the recommendations when you are dealing with a case of non-syndromic craniosynostosis. All non-syndromic cases of sagittal and or metopic craniosynostosis, first we should screen for the SMAD6 mutations. And then all cases of non-syndromic coronal craniosynostosis, we should see for TCF12 and TWIST1 mutations. Similarly, for sporadic non-syndromic cases negative for above testing, research efforts to identify the genetic causes of non-syndromic should be taken upon basically by whole exome sequencing. Again, for familial cases of midline midline craniosynostosis, screening for extremely rare mutations in SMAD6 is seen. If negative, then testing for other genes can be considered. And for cases of single suture craniosynostosis which present with other congenital anomalies that are not consistent with any known syndrome, we should go with cytogenetic microarray, because as I will show in further slides, microdeletions and duplications are common. Next slide. So again, the same genetic defect and syndromes are mentioned, especially including the common ones of the syndromic and the non-syndromic ones. Next slide. Now, chromosomal issues. As I told earlier, around 15% of patients with syndromic craniosynostosis show chromosomal rearrangements. These rearrangements include partial deletions of 7p, 9p, and 11q, as well as partial duplications of 5q, 13q, and 15q. Next slide. Hello. Yeah. Again, click. Click again. Yeah, basically we have shown here the typical presentation of a patient with FGFR2 mutation. They are around 98% of the cases and caused by the mutation shown here. Most of these have an autosomal dominant mode of inheritance, and most cases arise by sporadic mutation. Craniosynostosis involves multiple sutures, mid-phase underdevelopment with retrusion is there. Again, hypertelorism, exophthalmos, and other typical parrot beak appearance of the nose is there. Apart from that, other features can be there. Next slide. Yeah, next. Yeah. Basically, FGFR2 and 3 mutations are involved for Crouzon syndrome. FGFR3, it basically causes Crouzon syndrome with acanthosis nigricans. Again, the inheritance is autosomal dominant. Yeah, and craniosynostosis involves again multiple sutures can be there. The typical for this is hypertelorism. Exophthalmos is very typical. Big nose with a mid-phase underdevelopment is quite common. But again, digital anomalies are not that common for Crouzon syndrome. Next slide. Yeah, next. Yeah. Now again, this twist one, which causes non-syndromic craniosynostosis, again, it can have a uni or bilateral coronal suture synostosis is very common. Apart from that, the typical features of ptosis, hypertelorism, mid-phase retrusion, syndactyly can be there. Apart from that, duplication of the distal phalanx of the helix is very typical for this twist one. Next slide. Yeah, here we are showing the Pfeiffer syndrome. And again, type 1 is associated with mutations in FGFR1 and 2. Pfeiffer type 2 and 3 are associated with mutations in FGFR2. Again, it is autosomal dominant inheritance, and all cases of Pfeiffer are sporadic mutations. Next slide. Yeah, again, here the Muenke syndrome, again having autosomal dominant inheritance, and synostosis along with coronal sutures is common. Sensorineural hearing loss can be there, flattened cheekbones, and about 5% of affected individuals may have macrocephaly. Next slide. Yeah, again, this is the craniofrontonasal dysplasia caused by involvement of the EFNB1 gene. The coronal synostosis leading to brachycephaly is common. Facial asymmetry can be there. And basically a grooved nasal tip or a bifid nasal tip is typical of this disorder as shown in this figure. Now, next slide. Yeah, again, this shows Beers-Stevenson syndrome. Apart from craniosynostosis, cutis gyrata, acanthosis nigricans, skin furrows, skin tags, and anogenital anomalies are typical for this disorder. Next slide. Now, regarding surgery, of course, craniosynostosis, apart from diagnosis, the typical surgical involvement is there because of the cosmetic deformity and also for other causes of functional impairment like intracranial hypertension or optic atrophy. Next slide. Again, in this slide, I have shown the various surgical procedures which are being undertaken by the neurosurgeons. We can skip this to next slide. Yeah, the therapeutic trials. Lot of therapeutic trials are underway for these craniosynostosis disorders, especially the PD17304 of FGFR tyrosine kinase inhibitor has been used in a model of FGFR2 related Crouzon syndrome, demonstrating coronal suture patency. Wong et al. Have observed ligand-independent phosphorylation of FGFR2 in a model of Beer-Stevenson cutis gyrata syndrome and the P38 MAPK pathway activation. The use of such inhibitors suppressed the premature suture closure in mutated mice. Similarly, a TK inhibitor has been shown to rescue the major pathological effects of activating mutations in FGFR1, 2, and 3 in experimental models of chondrodysplasias and craniosynostosis. So basically, as for other genetic disorders, a lot of therapeutic trials are underway for such disorders. Now, basically, I moved away swiftly for giving a little brief introduction of craniosynostosis. Now, I would like to share two interesting cases with you all. Next slide. Yeah, this is a case one. Basically, this was a girl who was brought to our medical genetics department with complaints of poor growth, primary amenorrhea, and learning disability. She had a poor growth since birth and mild intellectual disability was present. Birth history was normal, developmental history was also normal, but again, she had poor scholastic performance. Next slide. Yeah, this is the pedigree analysis. As I told you earlier, because some certain residents are also part of this webinar, a 3-generation pedigree is very important, and the P is the proband which we have highlighted. Next slide. Again, anthropometry is very important for any genetic case, even for any pediatric case. We found that height was at -2 standard deviation, weight was at -2.5 standard deviation. Apart from that, occipitofrontal circumference was at -1 standard deviation. This is the typical IP growth chart on which we plotted the anthropometry of this girl, and we keep on seeing the trend as we followed up the child. Next slide. Yeah, on examination, one more request I would like to say to the participants: please don't click photographs of these patients, because they are we have sharing these photographs as a part of the consent. On examination, we couldn't find much in the child, but she had typical mid-face hypoplasia, flat occiput was there, raised heart ridge was there along the coronal sutures. Head to toe examination, no other abnormality was there, while general systemic examination were within normal limits. Her mother, we also found the mother was also having little bit of mid-face hypoplasia. Otherwise, she was not having any other complaint. Next slide. Now, regarding the investigations, karyotype was normal, ultrasound was normal. Basically, for any child with a little bit of intellectual disability or mental retardation, we do a thyroid profile, and we found in this case that it was suggestive of hypothyroidism. While a skeletal survey, we couldn't find any typical findings apart from the typical skull shape. Next slide. Yeah, basically.
You are on mute. Kindly unmute yourself.
Am I audible right now?
Yes, sir. Perfect.
Yeah, sorry. So basically, any case of craniosynostosis, a CT scan is must for a 3D reconstruction to see the extent of the craniosynostosis. And we found that this child was having brachycephaly, and the cephalic index was around 92%. What is cephalic index? It is the bipyramidal diameter divided by the occipitofrontal diameter into 100. Normal is between 74 to 83%. Next slide. Yeah, then we proceeded with this child with exome sequencing, and we found her to harbor a heterozygous mutation in the TCF12 gene, which causes craniosynostosis type 3, which is basically a non-syndromic craniosynostosis. As for any other variant detected on exome sequencing, we proceed with the ACMG guidelines, which were published in 2015. Next slide. Keep on clicking thrice. Again, click. Yeah. So basically, as per the ACMG criteria, we found this to be a pathogenic one, because protein length changing was there, and the computational evidence, the in silico analysis, also showed it to be pathogenic. Apart from that, it was absent in population databases. Next slide. Yeah, again, back to the here. Again, this is the same amino acid change as previously known pathogenic variant. And again, was a null variant in a gene where loss of function was a mechanism. And also, ClinVar was showing this as a pathogenic one. Next slide. Yeah, so for any genetic disorder, if any report comes, basically, I want the listeners to understand the use of online databases. Most of us must be knowing about OMIM, Online Mendelian Inheritance in Man. Basically, what it is, OMIM is a catalog of the various genotypes and genes and their different phenotypes or the clinical features with this they present. So when we put this TCF12 gene on the OMIM filter, we found the type of the disease caused by this gene and the various clinical features. Similarly, ClinVar is their database where we can see all the disease causing variants in different genes. It can be directly clicked from the OMIM panel. On the right side of the OMIM, you can see 1000 genome ClinVar exact, everything can be there. So you can directly go from there, or separately you can open ClinVar and see the various variants in a particular gene. Hello, am I audible?
Yes, sir, you are audible.
Yeah, next slide. So what are the take-home messages from the case one? Basically, this child was followed to us with a complaint of moderate intellectual disability, but during examination, we found her to have coronal synostosis. So learning message for the listeners is that if phenotypic evaluation is necessary, as in this case, the child presented with failure to thrive and amenorrhea were her main complaints, but she was found to have craniosynostosis on examination. Use of online databases is a must for proper understanding of the various genes, disorders, and rare diseases. And again, certainly a molecular diagnosis helps in delineating the etiology, management, and accurate genetic counseling. Now I will move on to the next case. Next slide. Yeah. Now this is case 2. This was again an interesting case. Basically, RK and his son was referred to us from the endomedicine department. He was being followed up for diabetes for last 10 years. But thankfully to the clinician there, the endocrinologist, he found some part of dysmorphology, some kind of dysmorphology in the patient, and the sons were having moderate intellectual disability, so he referred them to us. Next slide. Again, we same thing which we do for all genetic disorders. We draw a 3-generation pedigree in which the father, the 47-year-old proband, told his 2 sons have moderate intellectual disability and somewhat the dysmorphic features were common in all 3 individuals. And the mother of this proband, she died at 60 years of age with some gastrointestinal cancer, which exactly he was not having any documents. Next slide. Yeah, again, we took the vitals and anthropometry, in which we found that the son's height was at minus 3.28 SD, while father height was 147 centimeter, which was not that less. It was, but again, son's height was quite less. And again, the occipitofrontal circumference were almost normal. Next slide. Yeah, then on head to toe examination, interestingly, we found acrocephaly with facial asymmetry in both father and son. They were having a flat facial profile with prominent nose. Pterygia was noted along with hypertelorism, proptosis, dental crowding was there, large ears were there, and again, broad big toes was quite typical for them. Next slide. Yeah, here we have shown the clinical pictures of the father and his son. As you can see, the typical skull shape, acrocephaly in the father in the lateral view. Apart from that, you can see the duplicated big toe in both father and the son and the typical brachydactyly. Next slide. So again, this patient, the proband, the father underwent whole exome, and interestingly, on whole exome, 2 variants of uncertain significance were suggested by the testing laboratory. But again, they were not having all the features matching with the patients, and they were like partial phenotype match. So what we did, most of the time, the clinicians dealing with NGS and the geneticist must understand that generally when we don't get such typical findings, we should review the raw data of the NGS. So we asked the lab to provide us the raw data. Next slide. Hello. Yeah, next slide. Apart from those 2 VUS in which is a certain phenotype match was there, the lab also provided us 2 variant of uncertain significance for cancers. But again, this was because the mother was having a history of a GI cancer. But again, these were variants of uncertain significance. As you all, most of you must be knowing that as per ACMG guidelines, around 73 genes are there which cause cancer, cardiovascular disorders, etc., which we have to see. But again, only likely pathogenic and pathogenic variants should be reported. Next slide. So what we did, because most of the features in our patient were matching with the twist1 gene finding or the phenotype, that is the Sather-Cohen syndrome or the acrocephalopolysyndactyly, we asked the raw data from the lab, and we found a variant in the twist1 gene, which was a 21 base pair insertion as per the raw data. And on the figure which I've shown, it is basically IGV. It must be quite familiar with the people who are involved in the raw data analysis or the geneticist. We found that the twist1 gene coverage was also not good in the lab raw data. And basically, this was a non-frame shift insertion in exome sequencing raw data. And as per ACMG classification, this was VUS. And most of us, while filtering the various filters, while applying for the filtration of the raw data to get a significant variant, many times we ignore the non-frame shift deletions and insertions. So basically, this was filtered out. Next slide. What we did was we again took the CCDS sequence. CCDS, as you all know, it is the sequence of the nucleotides in the exomes of the any gene. And what we found that this 21 base pair was basically a same 21 base pairs were present before. So basically it was the duplication of those 21 base pairs. So basically this was not an insertion. This was a duplication from 397 to 417 and with the NMID which I have shown in this figure. Next slide. Then we did the pathogenic prediction. As we all know, we see the population databases while dealing with any NGS data. We also see in the disease databases and the prediction tools are there. What we found that this variant is absent in the population database, but as per ClinVar, this was pathogenic, reported many times before in patients with twist1 gene involvement. And it was also damaging as per the functional in silico tools like mutation tester and shift. Next slide. So yeah, again, the same ACMG classification, we found that this was pathogenic as per the ACMG criteria. Next slide. Yeah, so basically what is the take home message with this case 2? Most important thing is the clinical diagnosis. Detailed phenotypic evaluation has no alternatives. Like if we wouldn't have examined the patients, we would have never understood or seen the typical craniosynostosis present in the individuals. And especially for people who are dealing with raw data analysis, we just don't rely on filters. For any genetic diagnosis in NGS era, I made a code. F is first see the patient, I is images of the patient, L is lab data analysis and reanalysis, T is a targeted approach, like C for the genes which are matching with your phenotype. And again, evaluation of the results by yourself or in silico analysis is important. We should not blindly follow the lab, because many times error can be there by the procedure panels or by the human. Again, reanalysis and revision of report is very important, because every 6 months a variant which was VUS may become likely pathogenic or pathogenic if more studies come or functional support comes. And again, all these things, if you will follow, you will reach up to a successful diagnosis for a rare disorder. Next slide. Basically, in this slide, I have shown my mentors from SGPGI. This is Dr. Shubha Phadke ma'am, Sarita Agarwal ma'am, Kaushik sir, Dipti ma'am, and Amita ma'am. Basically, I dedicate this presentation to them because of these clinicians. I got an interest and started working on rare diseases. Thank you.
Thank you so much, sir. It was indeed a very informative and detailed slides with lots and lots of illustrations and images, which are like very useful to understand the case studies. And especially the pedigree analysis are of great help in understanding cases. So I think everybody had a wonderful learning experience through the webinar. So thank you so much, sir. And now I would like to welcome Dr. Ashish Fauzdar for the vote of thanks.
Good evening, all. Thank you so much. In the end, all good things come to an end. I would like to, on behalf of Radcliffe Genetics, I would like to extend my sincere thanks to Dr. Usha Dave ma'am for delivering such a, I would say, such an informative talk on rare disease associated with global development delay and intellectual disability. Ma'am, your talks are really insightful, and out of your decades of experience, it was very clear from your slides. Also, I would like to thank Dr. Mayank Nile for presenting and sharing his experience on cases of craniocytosis, for his excellent talk. Sir is very dynamic, very energetic, and we look forward to his guidance always. In the last, I would like to thank my management and marketing team, business and IT team, Himal and Ansh, for supporting this webinar to make this event very successful. We look forward to host you, ma'am, again in future. And thank you so much for this academic collaboration. Thank you all the attendees who joined us in this webinar on Rare Disease Day. And I think we celebrated this day in a very aptly with a talk from Dr. Usha Dave and Dr. Mayank. I think this would be the excellent way to present this Rare Disease Day. And it is also a Science Day. Let's also remember our great scientist, Sir C.V. Raman, who gave the Raman effect on this particular day. Thank you so much, ma'am. Thank you so much, Dr. Mayank.
Thank you. Thank you.
That's all from my side. Himal, do you want to add something?
Yes, sir. Thank you. I would like Dr. Usha Dave ma'am and Dr. Mayank Nile to kindly give their insights.
Yeah, what I would like to say is after Mayank's, and it's a very important message to the clinicians, and we also do that all the time. Just whatever from NGS you get those variants, you should not just accept as it is. Okay, they are bioinformatics people who do not know what is medical. They have not seen the patients. They do not know what is the different family history, etc. Though the important thing is you have to give all your clinical phenotypes, whatever all possible clinical clues to NGS, and one can come back also. And even VUS, even it may not be, as per the data which is given to you as the result by NGS, the laboratory, but it could be also relevant, to your patient or your case as a diagnosis. So always don't ignore the VUS, because it could be your first new rare case. You have to go back and do, which he had really nicely given the message. And I really appreciate, Mayank, that we also keep on doing the same exercise and then come to the conclusions. Thank you.
Apart from that, I would like to thank Radcliffe team and Dr. Ashish Fauzdar and Hemal and all those who arranged this webinar.
Yes,
That was a
yes.
very good experience, and it was managed quite well. And
Yes.
again, as Madam told, rare diseases are quite common nowadays, and because expertise is increasing, genetic tests are being taken up by many people. The cost constraints are decreasing every now and then. And apart from that, for the new clinicians and the other attendees, I would like them to know about the National Policy of Rare Diseases, which came last year, and the government is also ready to support treatment and research for these disorders. So basically, we should keep a track on these disorders, and on this special day, 20th Feb, which is celebrated for people with rare diseases, whether they are the clinicians or the patients or the labs, we all should work for a similar cause, so that people, basically, what is the intention of this webinar? The intention is to create awareness so that the proper diagnosis gets achieved for the patient and the doctor, and ultimately the life of people with rare diseases improves. And thanks again, thanks all of you for all the attendees for listening to us. Thank you.
Thank you, Dr. Usha Dave, ma'am. Thank you, Dr. Mayank.
Thank you, Radcliffe team and Dr.
Thank you so much, ma'am, for your valuable time. Thank you very, very
value
much.
your feedback. We look forward for your guidance in future too. Thank you so much.
Thank you, everyone.
Thank you. Bye.
Have a good evening. Bye-bye.